I'm supposed to identify the committee. So Brian Heisey from Natural Resource Science, Sorensen from Natural Resource Sciences, Keith Ferris from Natural Resource Sciences. And we'll have about a 15-minute presentation from Colin, and then we'll have questions from the committee and then from the audience. So I'll let Colin.
All right, so as you are probably all aware, I'm Colin Bailey, and I'm here to defend my honors thesis. and my thesis is on Chara species, marl, and trout, the effect of substrate and fish on benthic invertebrates in 6 Mile Lakes. So in BC, our freshwater fisheries economy is worth approximately $1 billion annually. And within Kamloops region itself, we're looking at about $100 million to the economy in terms of our small lakes. And what we do with these small lakes is we stock lots of rainbow trout in them, but we don't actually really understand the trophic impacts of stocking rainbow trout on these benthic invertebrates. And the reason why we care about these benthic invertebrates is because they form the basic food source for these stocked rainbow trout. Here is a pump sample, throat pump sample from a rainbow trout out of Mammoth Lake, and as you can see there are chironomids in there. This is a very common food source for these trout when these insects are hatching, and they can form a very large portion of the diet. So this issue with management has kind of come to a head recently because there's anecdotal evidence suggesting that a specific kind of weed bed called Chara in marl lakes has been declining, and that simultaneously benthic invertebrate populations in these lakes have been declining as well. So right here, this is Blue Lake, one of the lakes I sampled. This is a classic marl lake. It has nice Chara beds in there, which is this particular macroalgae right here. Grows like a plant, rooted in the bottom. And the reason why marl lakes are so important is because they produce some of the highest quality fisheries in the region. And by high quality, I mean they produce fish that are superior in eating quality to many other lakes. Now, part of the reason for this is these lakes are very high pH, very clear water. As a result of having a high pH, calcium carbonate precipitates, settles to the bottom, and forms the characteristic white bottom you see in these places. And in the process of this calcium carbonate precipitating out, it takes nutrients out of the water, particularly phosphate, and settles this to the bottom. And this increases water clarity because pelagic algae, phytoplankton, there's very little nutrients left in the water for them to grow. And this is what produces such high quality trout. So given the importance of these lakes, right, and the fact that Chara appears to have declined simultaneously along with benthic invertebrates, like this baited mayfly here, maybe they're linked. And what we need to look at now is, well, what is the importance of Chara, and what do trout have to do with this? So the main research questions here were, what is the importance of Chara? Does Chara provide higher abundance of benthic invertebrates? Does it support a higher abundance, higher biomass? And does it support a higher richness? You know, you would think that Chara, as opposed to plain white marl bottom, might produce greater living space, but also some more niches for different taxa to occupy. Another question is what happens when you throw trout into the mix? How do these communities change? What are the effects? And then, given that trout are size-selective predators, what happens to the large benthic invertebrates, the very large-bodied invertebrates, when you add rainbow trout? And what does this look like in a fishless lake versus a stocked lake? So we went out and we sampled 6 lakes, 3 stocked with rainbow trout only, and 3 fishless. So Lodgepole Lake, Unknown Lake number 5, or Lodgepole Lake, Gump Lake, and Blue Lake here were stocked with fish, and Unknown Lake number 2, number 5, and Shamrock Lake, they're all fishless lakes. Now, all these lakes were less than 16 hectares in size, and the reason for sampling such small lakes is because fishless lakes tend to be lakes that can't support fish over the winter. They winter kill because they're too shallow and often too productive. So in order to maximize comparability between lakes, the stock lakes that we sampled were very, very small. All these lakes were within 50 kilometers of Kamloops, and they're all very obviously marl lakes from the air. So when it went to these lakes, before you go, identified potential weed beds using aerial photographs. This is Gump Lake, one of the lakes I sampled. And then number these weed beds and randomly select one using an Excel random number generator. Then, upon getting to the lake, you'd go out to these to the care bed selected. First, you'd identify whether it was Cara on the bottom, or if it was a different weed bed, or maybe it was a rock shoal. So if there wasn't a care bed there, flip a coin to determine which way along the shoreline you travel, and then you would sample the first care bed encountered. If there was a care bed there, you'd set a 30-meter floating transect, as shown in this picture here, anchored in 2-meter depth water, and take 6 samples along there of the care bed using a petite pone arm. Samples were taken at random distances along the transect, again, randomly generated numbers using Excel. And then for sampling marl, so in the case of Gump, this was the weed bed that got sampled, and you can see marl around it, and there's actually marl in front of it as well. So reset the floating transect in the nearest adjacent marl that was also 2 meters depth. Again, took 6 samples randomly along the transect. Samples were then sieved in a 500 micron sieve, or a 0.5 millimeter square sieve size, and then placed into whirl packs with 95% ethanol to preserve them. Back in the lab, these sediments would be taken out, spread onto clear plastic trays, and searched twice under a magnifying lamp that's 4 times zoom, once over a dark surface like the lab bench here, black, and then once over a white tray with a white background to maximize your ability to find different colors of invertebrates and different darknesses. Then invertebrates were counted, sorted at the family level, and then biomass was determined per family and per sample. Here is one of the methods that was used for determining biomass, using a mass length regression. So length goes in here, and you're measuring this under a microscope using the ocular unit in the eyepiece. And then when samples were too numerous, so for example, chironomid samples where you had a ton of individuals, it just wasn't feasible to measure them all individually under the microscope. So they were dried for 24 hours at 60 degrees Celsius in a drying oven, and then weighed to determine dry mass. And then in terms of statistics, comparing overall abundance and biomass and richness between Cara and marl, and large invertebrates between stocked lakes and fishless lakes. These were all compared using Mann-Whitney U-tests, comparing means. And then for whole community comparisons, looking at changes in relative biomass and abundance, these were compared using ANOSIM, analysis of similarity, using sequential Bonferroni correction. And this told us whether communities were different or not. And then SIMPR was used afterwards to tell us what proportion each taxon contributed to the percent dissimilarity between communities. So here's the productivity and richness results. And what we can see here with biomass and abundance is that there is a significant difference between what Cara supports and what marl supports. Cara has significantly higher biomass and abundance overall of benthic invertebrates than marl. And then with richness, what we see is a similar relationship. We have more taxa on average on Cara beds than we do in the marl. However, there was no significant difference here, and this may have been due to the fact that marl was sampled immediately adjacent to Cara, which might have some spillover of taxa out of the weed bed. In terms of large invertebrates, generally what we see here is more large invertebrates and a higher biomass of larger invertebrates in Cara and in fishless lakes. The relationship appears to be a little clearer between stocked and fishless lakes in terms of abundance and biomass. With respect to abundance in Cara versus marl, we have one taxon here that isn't showing the same relationship. However, when you go down and look at the same biomass, we see that on average there's a little more biomass of probability than on the marl. These large invertebrates, these are the 3 largest invertebrates identified. Largest is in most biomass per individual on average. And there were no significant differences detected here, just fairly strong relationships from appearance. And in terms of Echinidae, which is a large dragonfly nymph. It was not detected in a single marl sample. And Dytiscidae, which is a large diving beetle, neither the larvae or the adults were detected in any of the stock lakes. Interesting result. Then when we look at whole community in terms of abundance, this is a non-metric multi-dimensional scaling plot here. And what this does is basically takes all the taxa and their abundances and then takes all these variables, configures it into a two-dimensional plot and shows you distance, sort of a relative distance between them. What we have here is these two overlapping polygons, Fishless Chara and Fishless Marl. They show a fair bit of overlap, although they were significantly different according to the ANOSIM. The most interesting part here is when you add rainbow trout, which is the red and green polygons here, what happens is that these differences seem to get magnified. So there were significant differences between all communities, but it appears that rainbow trout increased these differences. So here's the results of the SIMPR, which is telling us percent dissimilarity between groups. And what we have is Chironomidae, which is this guy right here, Sphaeridae, Gameridae, and Hyalalidae. Now, Chironomidae and these two amphipods are heavily fed on by fish, and it's interesting that they show up as as three of the largest contributors. Sphaeridae, it's I've never found any instance or reference about them eating clams, and they're very, very small and probably hard to find on the bottom. But they may be a competitor with something else that's affected by fish, or there's also the possibility, given that unknown lake number 2 had unusually high concentrations of them, there may be it may be just skewed the data. And then when we look at biomass, we see a very similar relationship here. Trout appear to increase the differences. And if you look in here, so these crossed points with the vertical line through them, there's only a couple that are really overlapping here, and then just one point that's sort of stretched out way in there. So really there is, and all communities were again significantly different from each other. So really we're seeing a very similar thing again here, but just not as, just a little more diffuse than the abundance results. And then if we have a look at the SIMPR, we see there's many more taxa contributing dissimilarity to the community in this case. And the reason why is because now you have these large invertebrates like Erpobdelidae, which is a big predaceous leech. Synagrionidae is a fairly good-sized damselfly, and Echinidae is this big dragonfly here. These animals are fairly low in abundance, but because they have higher biomass, they do actually contribute to community differences once you're looking at biomass. So conclusions, chara beds are more productive than marl. That's what it appears, that's what this looks like. And this is really very important because now we have a reason to actually go out and determine if there's any empirical evidence for chara decline. There's a reason to fund this now. And if chara has actually declined over the last 10 years, we should then start looking into research onto how to reverse these declines. And another important part from this is that we can actually maybe look into how to enhance these beds to enhance the productivity of these fisheries. Overall, it appears that chara beds and fishless lakes support more large invertebrates, but there's no statistical significant evidence here. So it's kind of up in the air, and this matches a few papers that have also suggested that while benthic invertebrates, the large ones, are decreased, the presence of weed beds actually mitigates for this, providing cover from fish. And then in terms of community, abundance, and biomass, really what we're seeing here are changes in a relative abundance and biomass of different taxa. We're seeing sort of a taxonomic shuffle, but we're not really observing the complete disappearance of large groups, at least in terms of families. Now, if we were to go down to species level, this might change. But on the larger scale, in terms of families, it appears that rainbow trout don't necessarily have as strong of an influence as we might have thought. And again, this might be because chara mitigates this community, the change in community. I'd like to acknowledge my supervisor, Dr. Brian Heisey, for his work and his patience. Tristan Tyler, Kaylee Overton, and Aaron Weeb for their assistance in the field. This was Tristan and I on our way to unknown lake number 5. Dr. Heath Garris and Jackie Sorensen for their excellent input and valuable advice with respect to this thesis. Dr. Nancy Flood for statistics advice, and Ministry of Forest, Lands, Natural Resource Operations, and TRU UREAP for providing the funding and making this possible. And then family and friends who have watched me lose sanity slowly and grow my mutton chop. So, any questions?
So we'll start with questions from the committee, and Heath, do you want to discuss?
Yeah, we'll start it off here. So you mentioned in your thesis, and obviously you mentioned here that you wanted to go back and do some sampling at different times of year. How would you anticipate, what sort of differences would you anticipate seeing in these communities? I know some of these invertebrates are more abundant at different times of year, but would you expect a wholesale different community or times of year when they're converging and you wouldn't expect to see any differences, say when fish are not very busy predating on?
So seasonally, like, and you'll see this in Ross's defense, the various prey that fish zero in on changes. And also with a lot of these aquatic insects, they have a life cycle they're completing, and part of that's outside the water. And so they have to rise and hatch, right? And so if you go and sample immediately after the hatch, and those animals haven't had a chance to breed, drop eggs back in, and then the next generation grow to a size large enough to actually detect here, because I was going with a 500 micron sieve size, right? You might detect less, and you will definitely detect less biomass. And so you might actually see different shifts, you know, in the relative abundance. I suspect you would still find almost all of the same players, but just in different abundances. And one thing to note with this is that when I went out and sampled, I found very, very few chironomid pupae. They were almost all in larval form, and this suggests that the bulk of the pupae that were going to hatch for the year had already done so. And so there were lots of larvae present, and whether those larvae were going to mature and hatch later, or whether they're going for a whole another winter before hatching the following spring, will have a large impact on what community you do find, right? Because if we're talking a 2-year life cycle or more, then we might have a pretty consistent composition of invertebrates on the bottom, versus if you're looking at something like mayflies, where they can get several cycles off in one season on a lake. If you sample immediately after, let's say, the first large hatch of the year, where those have been overwintering, it's going to be a little bit probably before you detect mayflies again in those benthic samples.
Cool. Good. I had another question, and this is more theoretical than suggesting you should do this work.
Yeah.
So you collected I don't know if you even told all these folks how many organisms you looked at. How many were there?
A little over 16,000.
There you go.
Yeah.
That's the sort of figure I love to see.
Yeah.
Okay, well, there weren't significant differences, but we tried. We definitely did a legitimate attempt. So I know you got a lot of these to the family level and probably some to genus and so forth, but imagine if you had all of these samples identified to the species level. What sort of information could you glean from that database?
So some of these families have feeding groups that are pretty much family specific. Like you look at dragon, well, even up to the order specific, right? Like dragonflies and damselflies, they're all predators, right?
Yeah.
But when you look at a group like chironomids, you got to go right down to at least genus or species quite often to determine whether that is a predator or a detritivore. They can encompass quite a few different feeding rules. So you could get some very, very interesting information with how the different feeding groups change at the species level. You would also, because I read a little bit in the literature about damselflies and how specifically there are some damselflies that colonize lakes and do well in lakes with no fish, and other species within the same family or even within the same genus that specialize in lakes that have fish in them. So I suspect you would start to see some differences there in terms of which species occurs where, and maybe even which genus occurs where. And the other thing is you probably start to see some more differences between Cara and Marl, because I noticed that there were tube-forming chironomids that specifically lived in the branches, in the sort of interstitial spaces of the branches. Their tubes were actually sort of adhering to these Cara parts, and then there were also tube dwellers in the substrate, you know, within the Cara, and then there were other things that weren't tube dwelling. So my guess is that there probably is a difference. And something else I mentioned in the thesis was with Cara and niches. The fact that when Cara dies off, you know, at the end of the growing season, and it's basically just sitting there, that's potentially a substrate that an algae that grows on hard surfaces could grow on. And in that case, that might provide a food source that isn't provided in a marl, where you have a loose substrate that's shifting. It's not very conducive to that kind of growth. So potentially there's another energy source there that might provide a niche space for different species.
Nice. Well, that led to my other question, which you just answered, which is, can we use the data we already collected to partition this data set into functional groups that you could then, you know, discern a trophic web and how that interaction changes? But
yeah, so quite a few of the families are you know, one functional feeding group, but then there's a few that are wild cards, like chironomids, and some of the caddis can serve in several different functional groups. So, but I think a lot of the main so we go back to in here, the Simper. Sphaeridae, like they're all filter feeders, right? Erpopdelidae, the big leech, they're predators. Hyalilidae and Gameridae, they're gatherer-collectors or detritivores. Chironomidae there's the wild card where you need to go to species.
yeah.
But Plenorbidae, which is a snail, that's a scraper. It's eating algae and various organic debris off the bottom. And then Cinegrionidae and Eschnidae, damsel and dragon, they're both obligate predators, so yeah, probably
So
could do some.
It's not totally resolved, but with a little bit of noise, you might be able to put together a trophic pyramid, especially
Yeah.
because it's biomass based, and you have some of these species, these families that are really just predators, or
Yeah.
you know,
Yeah.
that would be cool. You don't have to do that. Sorry, I just
I dove into it a little bit in there.
right. Yeah. Oh yeah.
Yeah.
And, yeah. Well, I don't want to take all the time. So,
First of all, I just want to comment on an excellent presentation.
Thanks.
And what a great job you did whittling down, 30-odd pages of tables and figures into a very concise results section, so we appreciate that.
Yeah, no problem.
I guess I'll start with the bigger question that came out of this for me, was that you provided, I think, really good evidence that Cara, you know, functions in this ecological role within Marl Lakes. But could other species of macrophytes that are tolerant to these conditions replace them and provide an equivalent role? And so did you think about looking at Cara versus other species, and how would you attempt to, I guess, explain that?
So there was one paper that I read, I think it was Hansen, and he was looking at what sounded like a marl lake in Alberta, but they never specified, they didn't tell me necessarily what the water chemistry was, and they didn't never label the lake as a marl lake. But he goes on to say that, you know, in the Cara beds that he was observing, there was a much higher density of dragonfly larvae in there than there were in, say, Potamogeton that was also in the lake. And quite often you do have other macrophyte groups in the lake, like Potamogeton. There's a freshwater moss that grows down there, which would be very interesting to test, because it at least in another lake, White Lake, it looked like this water moss occupied the next space down underneath the Cara bed, and who knows whether there's a shifting balance back and forth in terms of what depth they occupy. So that would be a very relevant thing to compare. And then Potamogeton patches and Cara patches occur in very similar depths often. And one of the main points of this guy, of Hansen, I think it was, is that this lake he was looking at had perch in it. And he said, you know, given the fact that Potawatomi does not provide a fish-proof barrier, but Cara seems to, you would think that if perch could access the dragonflies inside the Cara bed, that they would have that. They're opportunistic. They go for the largest piece of food they can get. So I would hypothesize that any of these macrophyte beds that aren't extremely dense or structurally hard like Cara, because Cara has a lot of calcium carbonate within the cells, that they would provide inferior cover and protection from fish for large invertebrates. For small invertebrates, having that just three-dimensional structure to sort of hide in would probably still increase productivity. And the fact that these, especially these softer plants, are probably providing a food source for some of these invertebrates, and they're definitely providing a carbon source for detritus. So on that front, they're probably similarly productive to Cara, particularly in the idea of sloughing cells to the bottom. They might actually produce more organic matter for benthic invertebrates to feed on.
So I guess from a fisheries perspective, then, does it matter whether it's Cara or Potawatomi? And you've answered that. Is it something that fisheries managers should be concerned about in terms of trying to enhance Cara populations, or is it okay if it gets replaced? But if you're talking about the disappearance of Cara and being replaced with a barren marl substrate, is that your experience of what's happening out there? Obviously, we haven't been to the lakes, so we're not seeing that.
So Blue Lake specifically, something that was very interesting to note was that I didn't see any other kind of weed bed. I saw littoral or like shoreline plants, right? But you had this band of white marl, and then you got to a band of Cara. I didn't see any other macrophytes around these Cara beds. There was a very tiny bit of something that looked like Myriophyllum near the launch, but it was very different, and there was no Cara there. And when I was sampling immediately adjacent to these Cara beds, a lot of that marl substrate was taken up by broken, dead pieces of Cara, like just small bits of Cara. And whether that's just pieces of Cara dying and falling off close by, or it could have been that Cara, it once inhabited that space, and when it dies and decomposes and finally falls apart, it forms marl substrate, and that's what's left over. And if that's the case, that's leaving a pretty sterile or relatively sterile environment left, and in a few of these other places, these Cara beds were basically kind of these sort of just islands, right?
Okay.
Yeah.
Thanks. I think that's something that I'd like to see maybe a little bit of clarification in your thesis in terms of why you're focusing in on this and ignoring other types of macrophyte beds.
Well, and the other part is that Cara Cara also enhances that nutrient sink in these lakes, which I wrote in there, and because the phosphate is binding to those Cara thalli, and Cara is providing the clear water, which is then providing a better quality fish in terms of eating. That's another reason to look at the importance of Cara, right? Is it's not only doing something for us in terms of benthic invertebrates, it's doing something in terms of the fishery quality that those other macrophytes aren't providing.
Should I defer to Brian and wait to the next round?
You can have one more question.
Okay.
It's like Christmas.
Just more of a technical question. This wasn't clear to me in terms of your method. I understand the complication involved when you have to mix regression-based biomass estimates with dry weights. So you use regression for most of your insect taxa, but for things like snails and leeches, it was more problematic. So how did you determine that threshold number of greater than 5 or 6 individuals? For me, it seemed very small, but was it just to deal with the error involved of using a scale, or can you comment on that?
So the scale I used went to the nearest 100th of a milligram, and I talked to Denise before going ahead and doing this, and she told me, I think it was, she needed approximately 4 ostracods, but the ostracods she was sampling, I'm pretty sure were fairly large, before she'd get any sort of measurable or reliable measurement out of the scale. And so really what it was from there was just an estimate, you know, like 25 chironomids, it's time consuming to measure them all under the microscope, but not deadly so. And once you exceed 25, you're approaching a biomass that should be measurable on the scale. With scuds and clams, because they're heavier, especially clams with a shell, you could go with a lower threshold number, just because these were things that you're fairly confident were going to come out with sufficient weight to actually be detectable by the scale. And I didn't go out and actually try to test exactly how many I needed. Instead, it was going with a safe estimate of, okay, once I'm at this number, I should for sure get a readable estimate. And the reason why is, I didn't want to destroy a sample by accident, or trying to weigh it in there and then not get a detectable weight.
Thank you.
Yeah.
Go ahead.
Oh, thanks. First of all, Colin, a general comment that I commend you for all the work you put in on this project. You came up with your sanity, as far as I can tell. And you, great job. There was a lot of hours at the microscope and sorting table. So I commend you for doing a great job doing all that. So, I understand you fly fish.
Yes.
And do you believe in using chironomids as bait?
No, I never used
fly?
them as live bait, but as a fly,
okay.
yes.
Sorry, it wasn't a trick
Absolutely.
question.
I am a diehard chironomid fisher.
So, do you fish with small chironomids?
Yeah.
Do you ever catch good fish?
Yeah.
Okay.
The largest fish I've caught is 8 pounds on a chironomid.
Wow, that's
8.5,
impressive.
yeah.
So you're saying that with tiny little chironomids, you can catch a really big fish.
Mhm.
Now, in your thesis, you say that trout are size selective and they like big food items. So I'd like you to reconcile those two things for me.
So, when you initially stock rainbow trout in the lake, the most likely thing that's going to happen is they're going to pick off the large, easy to grab targets. Like we saw that Dytiscidae wasn't detected in stock lakes, right? They're fairly slow moving. They have to go to the surface to get air and come back down. So you see probably a fair removal of these until there's some adjustment or equilibrium reached, right? The remainder of these, we saw there was generally more of these animals in the Cara than in the Marl, right? And if you're hiding in the Cara and you're adjusting your DL patterns of movement to stay hidden when you would be a visual target during daylight hours, because trout are visual predators, you can maintain some level of these large invertebrates, right, without the fish cleaning them all out. And then when you do have a hatch of something like chironomids, right, and there's sufficient biomass presented to make the risk of coming from the deeper zone where you're safe from loons and from eagles, there's enough of these individuals in the water to actually make the total biomass available very well worth your time to go through, swim through, mouth open, and just clean these invertebrates out as they're going towards the surface to hatch.
So are you saying that they're opportunistic as well then?
Yeah.
No? Okay, so you think those things are kind of balancing out? Are they to some
Exactly.
extent?
Yeah.
Okay, very good. If Steve Miracle is sitting in this room, he's not sitting behind me, is he, Steve?
No.
Okay, we have Jimmy. I want to make sure Steve's not here. If Steve Miracle is sitting behind me, what advice would you give him for how he should manage local trout lakes, given your results?
Well, I would first say that this is fairly preliminary and it only has 6 lakes in the study, and that I think it would be worthwhile to expand the size to start getting a sample size where, we're fairly confident that some of those Mann-Whitney U tests aren't hampered by a sheer lack of sample size. And then I would suggest that the next step is to try and find empirical evidence for Cara bed decline, and we've discussed how we might do this using aerial photographs and ground truth, and go out, use aerial photographs to identify where potential weed beds are, then go out, figure out whether they are Cara or not, and then take historical aerial photographs and using GIS or something similar like ImageJ, measure changes in area and look and see, okay, what's the pattern? Is it linked to climate? What might be driving this? If Cara has been declining, then the next step would be to research how to stop the decline, or whether there is any way to stop the decline, what's causing it. And then upon determining that, you then should have some idea of how to potentially enhance it. And given that, at least the family level, it didn't appear that, it looked like everything that occurred in the marl also occurred in the Cara. So if you were to enhance those Cara beds and actually increase their spread, theoretically right now, because we've only gone to family level, we shouldn't be removing habitat from something that specifically uses marl as opposed to Cara. So ethically, in terms of providing that diversity of habitats for your variety of invertebrates, you would think that increasing the size of the area in the lake that's inhabited by Cara would be beneficial productivity-wise, and it might be beneficial in terms of diversity, as we saw on average there was higher richness.
Would you go so far as to say that managers should take benthic production into account when stocking fish?
Yeah, because so there's some evidence from some papers that given the size selectivity of trout, that you actually get a lot of the larger chironomids apparently get eliminated, and the chironomid size goes smaller and smaller and smaller, over the years once you've introduced fish. And I would say that it's very worthwhile to potentially look at low density stocking, or so in the case of low density stocking, comparing it to high density stocking and seeing what the size classes are amongst all these groups, to see, okay, what's the difference in stocking level? What kind of different impact are we looking at in terms of our benthic invertebrates? If we stock at low density, does this have a lower impact, or is it the same? And it doesn't matter, and these trout are very good, very opportunistic, and you have less trout and larger trout applying the same effect. And then the other part to consider is once you get potentially a sample size of this study out into a larger group of lakes, and potentially down to genus or species, are there some species that are disappearing? How important are they? Should we consider putting lakes on rotation, where you stock them, you stock them for, let's say, a period of 5 years or 10 years. You let fish get to whatever size, they've got 5 years of good fishing life through there, and then you stop stocking and you leave that lake alone for a period of, let's say, another 5 or 10 years. Let the last round of stocking fish grow and mostly die off, give the invertebrates a chance to recolonize, increase, and then stock fish again. There's evidence like Phillips Lake where there was perch poisoning in there. They wrote it on the lake. Benthic productivity, anecdotally, according to the local fishermen, rebounded back seriously. I mean, we're talking massive increases in hatches again. And then when they stocked Fraser Valley catchables in there, the size these fish reached was impressive. And I haven't looked into whether they're stocking at a different density as the previous, but previously, before perch were introduced, the average size of the fish, according to anecdotal evidence from the fishermen, were smaller. Perch went in, the lake had a problem, they poisoned it, it got 1 year to recover. Apparently benthic invertebrates came back with a vengeance. And then when they stocked rainbow trout, it produced impressive sized fish. So by doing these rotations, we might create some very interesting fisheries where we have some very high productivity, and then those fish are basically removed, you let it recover, and then you stock it again. And that might also be beneficial for other things like amphibians, which tend to get pared down by trout because they are opportunistic and will eat tadpoles and things like that. So it could be...
That's a whole other argument. Isn't
Yeah,
it?
exactly.
I won't go there.
It could be beneficial for invertebrate diversity and for the fisheries as well.
Interesting ideas. I'll pass. Second.
We'll move back to the heat if you have any more
2
questions.
rounds?
Yep,
Okay,
do it.
I don't know. Lucky you. So how could you determine the appropriate number of lakes to sample to determine whether or not these differences you observed are statistically good?
We could do a power analysis. and determine, basically looking at the inherent variability of the system and determine how many lakes you need to start to find a measurable difference. And then once you've reached that, it's kind of like, well, if you add more lakes, you're probably not going to change the result beyond that point.
Yeah, and you can run that with the data that you have now. And
Yeah.
possibly put that as an addendum in your paper to say, hey, if you really want to know if these are significant, this is how much work.
Yeah.
So I have a very simple trophic question. So it looks like carra beds produce more invertebrates, more fish food.
Yep.
But they also protect fish food from being eaten.
Mhm.
So do lakes with more carra have more fish?
Well, so that's an interesting question because a lot of these lakes never had natural rainbows in them in the first place. They were introduced through stocking, and the population size is controlled by stocking. So the question there to ask or that I think should be asked is, do lakes with more carra cover produce larger fish per, or more biomass per fish stocked, right?
Right.
And some of the most famous, highly productive lakes in the area have very large carra bed coverage. And something of further interest is when I was sampling in the lab, it appeared that the denser the carra bed, the more invertebrates I found in there. So potentially, yes, more carra might equal more or bigger fish. But on the other side of things, having marl is potentially an important component because quite often, and this is just from being out and fly fishing, some of the best places to fish are where you have patchworks of carra patches and then white marl patches. And one of the reasons why this might be good is because it provides such a stark contrast. So when you have small chironomids rising up on a dark background of carra, they may not necessarily be as visible as when you have chironomids rising up and then just behind it you have this blinding white marl background. Provides a, potentially an excellent habitat for fish to prey on invertebrates. And the other thing is that if you have continuous carra bed around the entire lake, you may actually be reducing the potential for these fish to go and forage because when the invertebrates aren't hatching, and if they're not leaving the carra bed very often, you may actually be reducing the amount available to these fish at certain times of year. Now, they may just go down deeper and feed on gammarous scuds, amphipods. If you look in the Rawson paper that I've cited, he showed that at 10 to 20 meters depth, there's quite a few gammarous scuds, and quite often they're fairly large. There's a fair bit of biomass there, and they can contribute a very large portion of the rainbow trout diet. So it depends on exactly how much energy these fish are deriving from this carra-marl interface zone and what time of year before you decide whether you want a perfectly continuous marl or carra habitat or whether you prefer to try and create this patchwork to optimize feeding opportunity.
So that almost suggests that in some instances, carra decline may yield more fish because of introduced patches of marl within those lakes that are just chock full of carra.
So like White Lake has a continuous carra ring around pretty much the entire lake. And it's possible that having a patchwork might benefit this. However, to my knowledge, we don't really understand why carra forms an isolated bed like this or in strips with marl in between versus that continuous layer like that. So the decline of carra in White Lake might literally just be the bottom coming up or the top coming down in terms of the depth zone it can inhabit. And if it still remains as a continuous layer within that narrowed band, you're probably just losing overall productivity. If the carra died out in patches and you got these marl patches all through the carra, then you might see this positive increase in fish size.
How could
Jackie, do you have
I
any?
think you kind of answered one of my questions was that I didn't feel, I was convinced that these Marl Lakes could provide valuable fisheries, as you stated in your thesis, but I think you've convinced me.
Okay.
Another question about biomass. How are you on biomass?
Yeah.
Of all the time that you painstakingly went to measuring insects and invertebrates and also weighing them, do you think it was well worth your time looking at both abundance and biomass? And can you perhaps defend either one of those as being more valuable measures?
So I think it was extremely important to do both. And I think this reflects strongly in the SIMPR results I have in this presentation. So when you go up to abundance, the taxa show in there, studying these communities by abundance alone skews your results towards taxa that are very, very numerous, right? So only those that occur in really high numbers show up. On the other hand, looking at abundance is really good because if we're looking at, let's say, a bunch of chironomid larvae that are very early instar, they're all really, really small, but you know that later on they're going to become larger and they're going to present significant biomass for trout to eat. And this is where biomass comes in. Biomass is literally the amount of energy, or basically the amount of energy available for trout to eat, right? And so if we don't look at biomass, we're really ignoring the aspect of how much energy is available for fish to consume. So when you balance the two out, like when we look here and we see, Chironomidae provided 50% of the dissimilarity between communities. Wow, that seems very important. But when you go down to biomass, we're looking at, it's down halfway or a little below halfway. It has less importance. It's only providing 9% of the overall dissimilarity. If we were only to do biomass, we might think, okay, chironomids probably aren't as important, but having fly fish that have done all these throat pump samples and everything, we know that when these chironomids start coming off in numbers, the fish get in there and they get stuck. I mean, they're chironomids coming back out of their mouth. That's how many that are in them, right? So if we only looked at abundance, or if we only looked at biomass, we might think, but if we consider life cycle and timing and when they're pupae, they're at their maximum size, then abundance becomes important again. So I would say they're equally important to capture the whole picture, especially when you're only looking at a snapshot. If we're looking at all year round, maybe biomass would be better just because that is the actual energy available to those fish to eat.
Okay, and so having said that, would you consider running your analyses eliminating the most influential taxon?
In terms of abundance?
Both.
Oh, both.
See if your results change dramatically.
Yeah.
I'm not suggesting that you need to do this, but
Yeah.
it's something to think about.
So with abundance, if you were to do that, the problem you might run into is that, so when we look at biomass and see the taxa that did come in as significant that weren't in abundance, they're very, very large, but the density is so low that it might be really hard to actually find a significant relationship or detect a relationship in there. So if we had taxa that were abundant enough to provide enough basically sample size, I guess, to compare with, then yeah, it would be interesting to eliminate those top taxa and look at that. Because there's things like your mayflies, Betidae and Canidae, didn't show up in either of the SIMPR analyses. But Canidae, there were significant differences. There were significantly more canids in the Cara than in the Marl. But they're very, very small, and there's not quite enough of them to break into the abundance matrix. So yeah, there's some of those that would be interesting.
And chironomids and betids are, of course, they're famous for overwhelming results.
And trout really do zero in on betids when they start coming on.
Thank you, Colin. Brian?
One of the disadvantages of being last is that I'm a good question.
Yeah.
But I will add an addendum or a codicil to Jackie's question there about this abundance biomass. What's the best way to really get a handle on this? So my question would be, you had two ways of looking at the differences here in a multivariate sense. One was the diagrams. I really like the NMDS, nice, far apart, or together, I can grasp that.
Yeah,
And then you have things like this table, it's that total dissimilarity.
yeah.
What do you think is the best evidence? What do you think gives us most bang for our buck? What should the reader really focus on here to separate things?
Visually, the NMDS plots are the best.